and modifications. These are the modifications of the basic principles of genetics. So what were the basic principles of genetics? These were about the Mendel's laws, two laws. One was law of what was law of segregation and
law of dominance.
Yeah, law of segregation or law of dominance is same, yes, and the law of independent assortment, right?
Yeah. Yes.
So in law of dominance or law of segregation, we mentioned that there was a 3:1, right? Whenever we will have F1, all will be dominant, the character will be expressed, only dominant character will be expressed in F1. And in F2, there will be 3:1, right?
Yeah.
But there are some modifications also, like incomplete dominance or codominance. So some of the or the epistasis was there, right? Dominant epistasis and recessive epistasis. So here, like in Mendel's experiment, we find that all F1 plants which were heterozygous, for example, if there was a cross of capital T and capital T tall plants with the short plants,
Yes.
right, small t and small t. So the F1 phenotype was heterozygous, capital T and small t. So all it was heterozygous, but the dominant allele was more expressive there, right?
Yes.
Yes.
All tall.
All tall. And when the same plant, yani, the seeds of the heterozygous, capital T and capital T, when they are crossed, self-crossed, capital T and small t, in that case, we will have.
We got 1:2:1. That is.
Yeah. One is the pure homozygous
pure
dominant.
Oh, sorry. Yeah.
Yeah. And the other one is heterozygous. And,
yes. And one
third
is
one also heterozygous. So three are the tall phenotype and one with the short. But this is pure homozygous recessive, right?
Yes.
So there was one of the studies which was done by Lucien Cugnot, right? Cugnot in 1905. He studied the coat color in mice. The coat color means skin color in mice. So when he crossed yellow mouse with yellow mouse, so what could be the expected ratio? The expected ratio should have been 3 should have been yellow mouse and 1 should have been non-yellow mouse. If we see the Mendel's genetics, right?
Yes.
But he did not observe the expected ratio. He only found 2 yellow mice and 1 non-yellow mouse, and 2:1 instead of 3. So where that one ratio, where that one pure one went, then what did he do? He test crossed. Test cross means that whenever we have the heterozygous or unknown genotype, we have to cross it with pure homozygous recessive, yani small y. Any phenotype, genotype which is unknown, we have to cross it with the pure homozygous recessive parent, any small y. So non-yellow mouse was small y. Then we can get the genotypes and we can get the ratios. Then we can know what is its genotype. And then he found that the genotype in all these yellow mouse, it was capital Y and small y. So what is the general genotype? One should be capital Y, right? And 2 should be small capital Y and small y.
Small y and
yeah.
one small y.
2 will be that one, and 1 will be small y,
Small y, small.
small y. So he found these two ratios. He found these two, and he did not find that one, the capital Y and capital Y.
Okay.
So where did it go, this capital Y, capital Y? He observed that actually it was the same, like the one capital Y, 2 capital Y, small y, and 1 small y, small y. He found that this one also produced the yellow color, but at this stage, this capital Y mouse, it died during the development stage inside the womb of the mother. It did not develop. During development, it died. So when this one died, the offsprings were, which were produced, were 2:1.
2:1.
Yeah. Now, this color, capital Y, was dominant, right?
Yes.
And it should produce yellow color.
Mhm.
But why it died? Because it produced lethality. Capital Y was lethal. Not the mouse did not survive. And this capital Y combination, lethal means poisonous or bringing
Poisonous.
to die. Yeah. So
Yes.
so for development, for the coat color, skin color, it was dominant character. But for death, it was recessive character. So the mouse did not survive; it died. So this is known as lethal recessive allele. Lethal recessive allele. It is dominant for coat color, skin color, but it is not dominant for the development. For development, it was recessive. That's why this mouse, it died before the birth. YY character is dominant for coat color, but YY character is recessive for the development. And this is known as recessive lethal allele, right?
Yes.
Okay. So lethal allele definition is here, that it causes death. This is any allele that causes death at an early stage of development, often before the birth. So one of the genotypes, it will not appear among the progeny. So here is the cross, that a mouse, capital Y, a small y, heterozygous, and capital Y, small y. So what will be their gametes? Capital Y and small y gamete after meiosis, and capital Y and small y gamete after meiosis. Then these two gametes, by chance, they can meet any of these two gametes and fertilization will occur. So one of the capital Y, which was yellow, but it died in the embryonic stage, in the development stage. And the heterozygous conditions, half of from these four, yani two of these four, they were yellow colored, and one of them is the non-yellow mouse. So here is the same definition, that allele that causes the organism to die, these are called lethal alleles, and the genes, these are involved, these are essential genes. So here I have explained that capital Y and capital Y, this is in the video, you can see in the video. Capital Y and small y, and capital Y, small y. Capital Y died. Capital Y, small y, and capital Y, small y, these two were yellow coat color and they survived. And this one is non-yellow, small y, and it also survived. So the mice, they are homozygous for Y allele, they die. Therefore, the yellow allele is a lethal recessive. So it is recessive for development, but dominant allele for coat color.
Yes.
Next example is about the Snapdragon plant. It is also known as Antirrhinum. So Erwin Bohr, in 1907, he found the similar phenomena in plants. There, the first one example was about mouse in animals.
Yes. Animals.
The second example is about Aureus strain, where the lethal allele was present. In his first cross, the plants with green leaves, he crossed with the plants with the green leaves. So all plants were with the green leaves. But when he crossed a plants with yellow leaves and a plant with yellow leaves, so the result was 2 yellow-leaved plants and 1 green plant. So obviously, capital Y, the plants with yellow leaves, they did not survive. So what may be the reason that this capital Y did not survive? What is missing in the leaves? What?
It is also little. One died during.
No. Okay. So plants have leaves always, right?
Yes.
And these the green leaves, what is the function of the leaves?
Chlorophyll, this thing, photosynthesis.
Exactly, photosynthesis is their function. Chlorophyll are present.
Yeah.
And because of the
Okay.
chlorophyll molecule, light is reflected. And that's why they look like green. So when chlorophyll pigments are not there, which are responsible for photosynthesis, definitely the plant will die. They will be yellow in color, so they will die because the chlorophyll pigments are not there for the photosynthesis. So that's why that plant did not survive, though it was dominant for color, yellow color, but it was recessive for its, so it was lethal character for its development. So here, that one was the same example, that Aureus strain. Aureus strain. 2 plants were yellow, and one of them was green. When green is crossed, all progeny had green leaves. When yellow is crossed with the green, half of progeny were green leaves and half of the yellow leaves. So that confirmed that yellow-leaved snapdragons are heterozygous. So it was a test cross. The second one, when yellow is crossed with the green. So yellow is definitely capital Y and small y, then they knew that it is heterozygous. And when it is test crossed, so test cross means, sorry, not this one. Test cross means small y and small y, homozygous recessive. This is a test cross. So what will be the result? The result will be capital Y and one small y, and another also capital Y and one small y,
Small y.
and small y,
small y.
and small y,
And again one more small y.
again small y, and small y. If this is the condition, yani half of them are heterozygous yellow color and half of them are green. When they find like this one, so they found here this one, half green leaves and half plants with yellow leaves. Do you understand this one? When
yes,
they find this 2:2, yani 1:1, half, then it is confirmed that it is heterozygous after the test cross.
Okay.
Yeah. So, but if it is not heterozygous, this capital Y and small y, if it is capital Y and capital Y with small y and small y, there will be no green. There will be all of them, they will be capital Y and small y. All will be like the heterozygous, but there will be no green. If there is no green, then we can say, yes, it was pure homozygous dominant capital Y, capital Y. But if we find this ratio, 2:1, then we can know, yes, it is heterozygous, capital Y. So they found it that it is heterozygous. So again, the question came that where was that capital Y, where did it go? The capital Y and capital Y. So it was the same like mouse, that this plant did not survive, it died. So 2:1 is a recessive lethal allele. Understand?
Yes, sir.
Okay. Now, some other examples are there, multiple alleles. For some plants in genotypes, we always have the several alleles, more than one allele for the plant will be diploid, and they will have only two alleles in its chromosome, but they will have more alleles. Sometimes they will originally they will have two alleles, but more than two alleles are present, and the combination may be different. So one of the examples is they have more than two alleles, and that's why these are known as multiple alleles, or they are known as allelic series. But in that case there will be only two alleles will be present in the diploid organisms. So one of the examples is duck feather. So duck feather has three alleles, more than two. So one of them is allele M, which is mallard. Another one is allele MR, which is restricted. Third one is allele MD, M small d, which is dusky. So restricted is dominant over mallard pattern of the feathers. This is about the feather patterns and colors. And small m is the most recessive one. It is known as dusky. The dusky means dusky means the dark colored feathers. So how we can know how many genotypes if they have three alleles? How many genotypes they will have? So we can know by this formula: n plus 1 in the bracket, it is multiplied by n. Again, n is number of the alleles divided by 2. So if we have three alleles, so three alleles will give us 6, 1, 2, 3, 4, 5, and 6. 6 genotypes they will give us. How? Look at this one. Three alleles are there. In the bracket, we write 3, and then 3 plus 1. So how much it will be? And divided by 2. So 3 into 4.
Divided by 2.
So it will be divided by, it will be 12, right? So
Yes,
12 and divided by 2. So there will be 6 possible genotypes. So we have 6 possible genotypes. These 3 are dominant, restricted, because capital MR is present. If only one allele is present, capital MR, so all of them, they will be restricted. Doesn't matter, whatever allele is present here, right? If it is MR, M, or MD. So in case of mallard, capital M and capital M is present, and capital M and small md. Mallard is the second one, which will be dominant over MD. And pure homozygous recessive MD is dusky. So look at this one. This is the feather pattern in the restricted duck. Mallard duck has the green head and the little bit grayish hair, and the brownish is on their chests. When restricted, heterozygous, capital MR and MD is crossed with heterozygous mallard, the gametes are capital MR, small m and d, capital M and MD. Fertilization occur, so this gamete may fuse either with capital M, or the same gamete, either it may fuse with MD, or small md may fuse with capital M, or small md may fuse with MD, right? So one of them was restricted, two of them were restricted, and one was capital M mallard, and one was dusky. So the ratio was 2 ratio 1, ratio 1. So it is not like 3:1, which we observe in the law of segregation, law of dominance. Second example is about ABO blood group. In ABO blood group, A capital I and capital A allele is represented for A antigen. Capital IB is represented for B antigen. These both are dominant alleles. And small I, it has no antigens. It is for O blood group. Yeah. So A capital A is dominant over small I. Here it should be small I. And capital B is dominant over small I. And when I and B are together, it will show co-dominance character. Both will show themselves. That's why some people, they have AB blood group. So
Yes.
here is the A blood group. They have genotypes capital IA and another genotypes capital IA and small I. So antigen A is present and antibody B is present in them. Capital B phenotype has capital IB and IB, pure homozygous, or heterozygous IB and small I. They have antigen B and anti-A. AB blood group, the people with AB blood group, they have genotype of capital IA and IB. A and B antigens are present. They don't have any antibodies. Now, O has small I and small I, pure recessive. No antigens are present, but anti-A and anti-B are present. A can donate blood to AB, A can donate blood to B, A can donate blood to A. But
Oh,
sorry, O can donate blood to AB, O can donate blood to B, and O can donate blood to A. But O
but
cannot receive, yes. It cannot receive, cannot accept any blood from A.
yeah, just O.
Just O it will receive, yes.
Okay.
So this was one of, okay, here, if a person, I explained in my lecture, you can see that one of the famous, it had practical example. Charlie Chaplin, you know Charlie Chaplin?
Yes, sir.
Charlie Chaplin was a famous comedian actor, right?
Comedian. Yes.
Yes. And he had a blood group, sorry, one of his partners, she went to court and asked for the expenses of the daily expenses for his child, and she claimed that this child belongs to Charlie Chaplin, and they had broken, they were separated. So the blood of the Charlie Chaplin was tested, the blood of that lady was tested, and the blood of the child was tested. So just in that case, what happened, that the blood of the child was different. So here I will just make an example of that one. So okay, maybe I have somewhere. I will go back to the Charlie Chaplin example here. Multiple alleles, where is that one? Yeah. So here you can see that the child had B blood group and Charlie Chaplin has O blood group. When someone has O blood group, the allele, it is pure recessive, small I and small I, right?
Yes, sir.
And Berry, she had A blood group. So A blood group, one of the possibilities is capital A, IA, capital I, capital A. Another possibility is capital I and small a, right?
Yes, sir.
So in first case, capital IA, capital IA, pure homozygous.
Okay.
So what will be the F1? All the children, possible children, they will have capital IA and small I. means A blood group, right?
Yes, sir.
If it is heterozygous condition, capital I, A and small I, small I. So in F1, here is an F1. Heterozygous IA and small I, and homozygous O blood group. This is the Chaplin blood group, Charlie Chaplin. Now, what will be the combination? Capital IA, small I is A blood group. Capital IA and small I is A blood group. Again A. And
This.
small I and small I. O. So half of the children, expected children, they will have A blood group. Half of the expected children, they will have O blood group. So what was the child blood group? B. So we are, in this case, what do you think? Does it fit here? This blood group?
No.
No. So when this, it was for the first time, it was the blood groups were tested for the in such cases that a very famous person's blood group was tested. So the child that did not belong to. So this blood group testing showed that child does not belong to
Charlie.
Charlie Chaplin.
Oh, Chaplin.
And then yes, Charlie
Yes.
Chaplin. Yes. So this was the AB blood group. And then we will have another example of the coat colors in the rabbits. So one of the colors of the coat color of the rabbits is brown, which is known as full color, and it is represented by capital C. Wherever this capital C will be present, either it is in homozygous condition like this one, it will be always dominant. And the other color is chinchilla. It has black tipped white fur. And third one is Himalayan. So here is the full brown chinchilla color. They will have something yellowish spots will be there on the skin. And one is Himalayan. Himalayan, you can see they have the yellow, the black ears,
Yes.
black nose tips, and black feet, and the little bit tail will also be black color. So this is Himalaya color. So full color is dominant over chinchilla, over Himalaya, and albino. Albino means pure white. There will be no melanin pigment. So no skin color pigment will be present. So full color is dominant over chinchilla, Himalaya, albino. Chinchilla is dominant over Himalaya and albino, and Himalaya is dominant only on albino. In full color, capital C will be present. All the genotypes will be, this is capital C. All the genotypes will be full color, brown. Doesn't matter.
Sir.
Yeah.
What is dominant over what? I didn't hear that.
This capital C full color genotype is dominant over all these colors. Whenever this capital C full color brown is present, whenever this C full color dominant is present, doesn't matter whatever genotype is here, your chinchilla genotype is here, allele is there, or Himalayan allele is there, or small c for albino allele is there. But if capital C is present, all the progenies, they will show brown phenotypes. Yes, full color phenotype. In case of chinchilla, capital C and small CH, if this allele is present, and they have chinchilla or Himalaya or small c for albino, or if any of these allele, the second allele is present, the coat color will be chinchilla. But when chinchilla is with capital C, so the capital C will be dominant. Full color will be dominant. Himalaya, which has the fur with the black paw, nose and ear tails, this is only dominant over albino. So yeah, either pure homozygous Himalaya or heterozygous with the small c. So the progenies or the rabbits will, these genotypes will have the phenotype of the Himalaya, and albino is small c and small c. So this is the about the multiple alleles. So Here I will discard this one. This was one of them. So I explained already this color. Now the third one is about the gene interaction in capsicum pepper, capsicum annuum.
So that is fourth example, right?
Sorry?
In multiple allele, fourth example.
Multiple alleles, we had 3 examples.
Okay, this is a different topic.
This is the 2 genes now, they are interacting with each other. Capital Y, capital, this is codominant character. Capital Y, capital C, capital C. So when this pure homozygous dominant 2 genes are there, the pepper color will be red. And pure homozygous recessive, small y, small c, it will have creamy color. So now 2 genes are there, not 1 gene. So 2 genes are for the fruit color. You understand? It's not allele, not 1 allele. 2 genes
Yes,
with
sir.
4 alleles. When they are crossed,
Yes,
so capital Y and capital C gene and small y and small c gene. So what will be the result? Heterozygous, capital Y, capital C, small c. But it will be red because capital Y and capital C are present. Now,
okay.
when this the seeds of capital Y, capital C, they were grown, and the plant was self-crossed, the pollen grains of the same plant, they were sprinkled on the carpel of the same plant. Yani self-pollination was carried out. So this cross showed capital Y. 9 of them were capital Y and capital C. Doesn't matter whatever genotype was, allele was there. So capital Y, capital C produced red. But when there was only capital Y, either capital Y or capital Y, small y, and other was small y, it produced the phenotype was peach in color. And capital C alone, and they had the pure homozygous recessive for small y. So when only capital C was there, orange was produced. And then the cream color was there because it was pure homozygous recessive. Now 2 genes, they are interacting for the color, for the fruit color. So 9331 was there, but 2 genes were there. So this is known as gene interaction. Is it clear?
Yes, sir.
Okay. Now we go to the another 2 explanations or 2 terms, penetrance and expressivity. So what is the penetrance? Penetrance is, where is my marker? It is here. Penetrance is the probability of a gene or trait being expressed, while the expressivity is the variation in phenotypic expression when the allele is fully penetrant. And example is polydactyly. Look at the fingers. So 1, 2, 3, 4,
6.
5, and 6 fingers. Polydactyly. Poly means many. Dactyly means fingers. So many fingers are there. So generally, a penetrance is described for the clinical expression, whether or not the clinical expression is of the genotype is there or not. So penetrance in genetics is the proportion of individual that carries a particular variant of a gene and that expresses an associated trait or phenotype. Penetrance is defined as the percentage of individual organisms of a particular gene that express. Now here is an example. If there are 42 people with the allele of the polydactyly and 38 of them, they showed the phenotypic character of polydactyly. while 4 did not show the polydactyly character, though allele was present. So this is known as complete penetrance. So it will be around 90% penetrance. 38 divided by 42 into 100. So it will be 90% penetrance will be there, or 0.9 penetrance is there from 1. Expressivity, it is the term that describes differences when between the two individuals, but the two individuals, they will have same genotype. Here is the same genotype may be there, for example polydactyly, but this polydactyly they may have the differences also. So in the clinical phenotype they will show differences. So the polydactyly is the degree to which a character is expressed. Yani some polydactylous persons, they have extra fingers and toes that are functional, but others possess only a small tag of extra skin. So this is the variation. Same genotype is present, but some, they have the extra finger and toe and they are functional. And in other persons, they have the same genotype, but they just show the extra skin. So this is known as expressivity. There is also another term, incomplete penetrance. Incomplete penetrance, when the genotypes, they do not show us the expected phenotypes. And what should be expected phenotype? 3:1. When we don't see expected phenotype, this is known as incomplete penetrance. So human polydactyly is the condition of extra fingers and toes, and this is caused by the dominant allele. Yeah, some people, they have the allele is there, but their fingers are normal, but their children, they will show their as inheritance, they will have the polydactyly. So this is it means that the polydactyly is not fully penetrant. So in one generation it did not show, but in next generation it showed themselves. So this is about the incomplete penetrance. And this incomplete penetrance or expressivity, variable expressivity, may be due to the effects of the genes, or may be due to the environmental factors. And the genes or environmental factors, they can change or they can suppress the effect of a particular gene. So this was the whole lecture about the variations, right?
Yes, sir.
Now if you have any question, you can ask.